Good afternoon, everybody. I'd like to welcome you all to this first Retina Australia webinar for 2022. It's a research update from two of our currently funded grant recipients, Professor Alex Hewitt and Associate Professor Fred Chen. My name is Sally Turnbull and I'm the Administrative Officer for Retina Australia, and I'll be the facilitator of this webinar today. Our first speaker today is Professor Alex Hewitt, who is a Principal Research Fellow at the Menzies Institute for Medical Research at the University of Tasmania, as well as a Principal Investigator at the Centre for Eye Research Australia. His major research interest lies in the ophthalmic applications of stem cell and gene editing technology. Alex obtained his PhD from Flinders University of South Australia in 2009 and completed formal ophthalmology training at the Royal Victorian Eye and Ear Hospital. Alex led the first report of viral-mediated Crispr-Cas editing in the retina. Today's talk is titled Prime Time for Inherited Retinal Disease Gene Editing. So I'll hand over to him now.
So yeah, thanks a lot for the introduction. And it's great to have the opportunity to talk about some of the research that we've been doing over the last couple of years, and principally over the last 12 months with the support from Retina Australia. In a way, I sort of think maybe Fred and I should have had our talks around the other way, because the major sort of thrust of our work has been on actually the genetic correction of variants that cause inherited retinal diseases. And I think part of Fred's talk at least will be in identifying patients who are, the genetic variants in different patients. So I guess I think when I think about sort of the changes that are happening in medicine and that will affect humanity, I think that Earthrise, has to be one of the most inspiring photographs for all of humanity. I find it amazing that we can send a rocket up into space, have that land on an asteroid, take a sample from that asteroid, and then travel back to Earth with a sample from that asteroid. I also find it amazing that we can launch a rocket into space that's 15 stories high and actually manage to land that back on Earth on a small platform floating in the ocean. And so, with all of these major sort of advances in physics, why hasn't biology caught up? And why isn't it possible for us just to go in and readily treat all of these inherited eye diseases? And I think in part part of the sort of answer to that is the actual complexity of the human genome. So your genome is comprised of 3.3 billion nucleotides or letters that make up your genetic code. And just to comprehend the size of that genetic code, if you imagine expanding each of the letters that comprise your genetic code up to be the size of a matchstick, and then if you stack each of those matchsticks end on end, you'd have enough matchsticks to stretch from the Earth to the moon and back to the Earth and then back up to the moon again. So certainly, 3.3 billion nucleotides is a lot. You can also then sort of think of your genetic code being comprised of one of 4 different letters. So essentially that means that you could color each of these matchsticks one of 4 different colors. And all it takes for a disease to manifest is essentially to have one of those matchsticks either burnt or just discolored, so painted one of the wrong colors. And that can happen just in one site, or for some conditions, you actually need a handful of different changes to occur. So in part, the difficulty in understanding or in treating inherited eye diseases has been in part just due to the complexity and size of the genome. Now, when we think about potential treatments for different inherited eye diseases, I think it's important to appreciate that the treatment that will be best is going to be very dependent upon how far advanced different people's disease is. such that the more advanced it is, treatments such as stem cell replacement and external technology are going to come to the fore, whereas for the gene editing and gene replacement therapies, which our team has primarily been focused on, is certainly going to be needed in the earlier stages of the disease. So essentially you'll need a reasonable amount of healthy remaining retinal tissue for gene editing to be potentially beneficial. So focusing on gene editing in particular, there have been recent advances over the past 8 to 9 years, and these were essentially spawned from discoveries from Emmanuel Charpentier and Jennifer Doudna's laboratory, whereby they were looking at the adaptive immune system of bacteria. And they found that there was a unique protein complex called Crispr, which stands for clustered regularly interspersed short palindromic repeats, that appeared to be appearing in the genome of bacteria. And they found that you could adapt this protein with an RNA complex such that it could then be used to target any specific region in the genome. So essentially, this protein is comprised of a protein component as well as an RNA guide, and that RNA essentially is what's used to bounce along the genome, and once it finds a match in a particular site in the genome, the protein is activated a bit like scissors, and the DNA is cut. Now, cells don't like its DNA being cut, and if you sort of imagine it as being a bit like the rope for a rock climber, obviously if the weight of the rock climber's weight is on the rope and you cut the rope, they would naturally fall. And similarly, if DNA in a cell is cut, the cell will die. So cells, mammalian cells in particular, have evolved mechanisms to rapidly repair the DNA if it is cut. And essentially, rather than repairing that in a precise manner, the cell's mechanisms just aim to join the DNA back together as quickly as possible. And unfortunately, errors can occur when that happens. Now, as was mentioned before, our team was the first to actually apply this gene editing technology via a viral mechanism to the retina of a preclinical model. And essentially what we showed was that with very high efficiency, you could actually use this machinery to alter the genes expressed in the retina. So that's all good and well if we want to potentially remove a gene or stop it from functioning well. What about if we actually wanted to correct a specific disease-causing variant? And so on that front, there have been some major advances. Primarily that started again with this Crispr enzyme complex, whereby two groups in the US and in Japan appended an additional enzyme onto this Crispr endonuclease. And essentially that enabled, they also blunted the scissors on one aspect, and essentially that enabled the chemical conversion of one nucleotide into another. And essentially that means that you could then convert a blue matchstick into a red matchstick or a black matchstick into a red matchstick. The following year, after that major advance, a different enzyme was evolved and appended onto the endonuclease, and that essentially facilitates the conversion of a green matchstick into a black matchstick or a red matchstick into a blue matchstick. However, what was alarming with all of this is that it was subsequently reported that as this complex was bouncing through the cell, it was inadvertently editing other sites. So it wasn't just going to the site that you were aiming for it to target. It was, as it was transferring around, it was having functions on other genes. And essentially, one way that you could think about this would be that if the instructions or if the genetic code in the cells, what you're hoping to have it say was, say, cook the lemon cake. If there was a spelling mistake that actually whereby the instructions were cool the lemon cake, obviously if you're going to aim to send in a Crispr editor to correct the L in cool to make it a K in cook, what was happening is that you were inadvertently getting other spelling mistakes introduced. So essentially that's a bit like saying, cook the Kemen cake. So if that hopefully sort of makes sense. The one sort of big analogy for these CRISPR enzymes and the base editor system is essentially it's a bit like having an eraser and a pencil combined in one. And we had a talented PhD student who, based on some insight from David Savage's lab, realized that you could essentially hide that eraser inside the pencil. And that meant that it is possible rather than having it just tagged on one end of the protein and therefore having that enzyme just waving in the wind, so to speak, you could essentially bury it inside the protein, hide it from other proteins, and actually bring it closer to the DNA of where it could have its function. And so essentially what Peter did was actually screen through a range of different variants, and essentially he found a sweet spot as to where you could hide that eraser. And then from there, he went on and screened a number of different variants and with the aim of correcting those genetic changes. And he found that the hiding the eraser inside the pencil actually increased the efficiency across a range of sites and also dramatically reduced these off-target effects. Peter went on to show that it was possible to package all of that system up into an adeno-associated virus, and that could well be important for future therapeutic delivery. And he also went on to show that his engineered CRISPR system was actually the most efficient platform for the genetic correction of a individual change that causes Usher syndrome. So in particular, a variant in the protocoherin 15 gene. So essentially going back to that analogy of where the instructions were to cook the lemon cake, essentially it meant that we had adequately corrected that initial spelling mistake, but also avoided the inadvertent off-target effect. So the natural question that leads on from all of that work is that, well, what about other potential disease-causing variants? So all of that technology is good if you just have one of those 4 specific changes in that particular sweet spot. But what about all of the other range of genetic variants that can cause inherited disease or inherited retinal diseases? And so returning to that CRISPR nuclease complex, David Liu's lab in the US actually came up with the novel idea of appending a different enzyme onto a different part of the CRISPR complex, and also extending that RNA template that's actually used to guide that protein along the genetic code. And essentially that technology allows the genetic conversion of a green matchstick into any other color matchstick, and similarly a black matchstick into any other color, a blue matchstick into any other color, and a red matchstick into any other color. So essentially they showed that it was possible to correct the genetic change of essentially any small genetic change that causes a range of disease. So the big question now was, how does this transfer across the range of genetic variants that cause inherited eye disease? And so in an attempt to address this, we designed essentially a big pooled experiment. And the big issue with this technology is that there's a range of different combinations that you can have to essentially optimize the system. So in order to get as high editing efficiency as possible, it's necessary to make a number of tweaks to the RNA and protein complex. And it's not initially clear as to which one would lead to the most effective designed a template whereby the repair template as well as the target sequence was appended to each other. And this was essentially designed for close to 12,000 established disease-causing variants that cause a range of inherited eye diseases. And what was exciting was that we found that using this approach, we could definitely identify distinct patterns of genetic correction. And essentially that allows us to tease out some important factors that must be considered when designing these genetic correction therapies. In particular, that we found that the distance between where the scissors are activating and the length of that repair template is particularly important. So when it comes to the clinical translation of this technology, I think we're certainly edging closer to the clinic. As Fred will introduce in the next talk, certainly the genetic diagnosis of inherited diseases has improved dramatically over the past decade. It's now possible for us to design the things and tools that will correct these genetic changes if they're well defined and in a small section of the genome. It's possible for us to model these conditions, so such that it's possible to test this out in the laboratory before actually applying it to an individual. And then finally, across Australia now, there's a range of different improvements that are happening that will ensure the production of these therapies. So I'm certainly grateful to the small army of PhD students and postdoctoral fellows and other collaborators who've contributed to all of this work. And I think one of the big take-home messages, hopefully, from the talks today is that there certainly is a small army of people who are working very hard on all of this. And finally, we'd like to thank all of the funding support that's contributed to this work, in particular the grants from Retina Australia. And I think that's it's really important to highlight the role that these grants do have in leveraging ongoing and bigger funding. So I'd certainly, I think, happy to open the floor now to questions or have a combined questions at the end. And if people are shy, you're certainly welcome to put questions into the chat.
Okay, Reeks van Klinken.
Oh, hi, g'day, Alex. Great presentation. I was just looking recently at a paper by, I think Lee et al. A couple of years ago that reviewed a lot of these genetic technologies and some of the issues around getting them working in practice. And I left that a bit depressed about all the barriers. What's your feeling in terms of, you know, the practical issues that have to be overcome to actually get this working in a sort of cost-effective way?
Yeah, so I think we're damn close, and like, you know, always with research, it's, you know, 2 steps forward, 1 step back. I think, you know, so I was saying in terms of gene editing technology in the eye, there has been a trial started for Leber's congenital amaurosis, so a rare condition that primarily affects children. And that is just the sort of scissor technique, not the actual genetic correction or base editing method. But there are clinical trials about to commence, and there's actually one starting in New Zealand in a few months where they're actually aiming to apply that the base editing technique to the liver and to correct for hyperlipidemia. So I think, you know, the barriers are rapidly being overcome. So I'd be certainly more optimistic than pessimistic about it. The big issue, you know, will be, I think, intervening at a time when patients would actually benefit from the therapy, where otherwise they'll need sort of other sort of modalities that would be needed. So hopefully that sort of answers the question, Rex.
Thanks. Yeah, I noticed you're actually an author on that paper as well a couple of years ago.
Yeah. I think, again, with all of these technologies, you never want to be first in line, but you don't want to miss out. But I think if there's been anything good come from the COVID vaccine stuff, I think it's the proof that, you know, these platforms can be scaled up for therapies. So I'd certainly be hopeful and bullish that these therapies could be rolled out in a manner that won't break our healthcare system.
Thank you so much, Alex. And I think now that Fred is with us, we'll go ahead with the next speaker. So Associate Professor Fred Chen is an inherited retinal disease specialist at the Lions Eye Institute and Royal Perth Hospital. His academic position is at the University of Western Australia and the University of Melbourne. Fred conducts several clinical trials in retinal diseases as the principal investigator and is the head of the Ocular Tissue Engineering Laboratory, which focuses on translational research and drug development for genetic eye diseases. Fred and his group have published over 200 peer-reviewed papers, and he serves on the editorial board of international journals and advisory boards for several pharma companies that develop novel treatments for retinal diseases. He's currently an investigator for treatment trials in Stargardt disease and Usher syndrome. Today, he's going to be talking about looking for disease-causing mutations in families with dominant RP pedigrees. Thanks very much, Fred.
Thank you, Sally. Thanks for introduction. And it's a pleasure to present on our research funded by Retina Australia. I'm going to talk on dominant RP. And first I'm going to touch on inheritance patterns, because there are confusions regarding how we call dominant, recessive, and Xlinked. Then I'm going to talk about the genetic testing that were performed in these dominant RP pedigrees. And then I'm going to talk about treatment for PRPF31, which is one form of dominant RP. So where do our patients come from? There are two sources of patients. My patients are enrolled into what we call the WARD study, which is Western Australian Retinal Degeneration Study. Majority of these patients are from our states, and I have the impression that it's about 50% of all the IRD patients in the states are in this registry. There's also the Australian Inherited Retinal Disease Registry, led by John DeRoche. It's a national registry, and they suspect that they've captured about a quarter of all the Australian IRD patients. I talked on this webinar 2 years ago, in May 2020. And at that stage, I presented how our WARD study was tracking, and we recruited 800 patients up to 2020. And in the last couple of years, we've recruited more. So there's now over 1,150 patients. Not all of these patients are or have inherited retinal disease. There were 684, so just over half have inherited retinal diseases as of May this year. About 300 have rod cone dystrophy, or we commonly call it RP. 294 were typical RP, so these are bone speakers with vessel attenuation, pale discs. Some of them have asymmetrical RP, 5 of them called sector RP, and 8 had typical features of choroideremia. There were also patients who are female carriers, and we know they have carrier status because of the way the fundus look. 8 of them were female carriers for choroideremia, and 17 were for X-linked RP. So that's RPGR and RP2. So I'm going to focus on dominant RP. Now, when we define this, it seems quite straightforward for most clinicians. What's X-linked, recessive, and dominant? There's a group of patients where we don't know which one it is. So we call them simplex because they're the only person in their family who has the disease. There's also mitochondrial diseases, which I'm not going to talk about, but these are inherited through the mother to all the children, and only females can pass it on. It doesn't typically cause RP, they cause a mitochondrial retinopathy, which can be confused with RP. KSS is Kunsaya syndrome, which is a condition with the motility disorder, so problem with moving the eye, plus retinopathy. So I won't talk about that group. I'll focus on X-linked, recessive, dominant, and the simplex. So as you can see, quite a large proportion of patients are the only person in their family affected by this condition. That's 42%. So what determines this kind of inheritance? Well, we have to look back to understand what the gene is. Gene, I tend to use the example of Lego blocks. We have about 29,000 genes on the DNA, and each one of these genes is a bit like the instruction booklet in the Lego box. It tells you what to do with these blocks, and you can make a little pattern or a little toy out of it. We have 2 copies of every instruction booklet, one from the mum and one from the dad, except for those instructions on the sex chromosomes, so the X and Ys. The cells uses both copies to make protein. So these are like the Lego blocks that are built. These can be enzymes, growth factors, receptors, structural proteins, and many other things. When there's a faulty copy of the gene due to errors in the code or missing codes altogether, there can be loss of function because the protein is not made, or the protein doesn't work when it's made. Alternatively, it can lead to a production of a protein that is altered in its function, which then inhibits other proteins made from the normal copy. It can also lead to excessive production or hyperactivity of the protein. So enzymes can be more active, for example, or there could be more of the growth factors produced rather than less. So there's three things that can result from faulty copy of the gene. The simplest inheritance to understand is actually the X-linked inheritance. So males have only 1 copy of the X chromosome and 1 copy of the Y chromosome. Females have 2 copies. But only one of these is functioning at any one time. So males will manifest disease when the gene on the X chromosome is faulty because there's no backup. The females will manifest retinal disease when most of the retinal cells inactivate the normal X chromosome. We still don't know really how female manifest these diseases. There's research being done here and in Melbourne looking at why do carriers manifest disease in some people. So we do see choroideremic carriers and RPGR carriers who are severely affected by the condition and others are not affected at all. Here's a family tree. It's rather complicated, but you can see the females are drawn with the dots inside, so they're carriers, and the males, the boxes here are the affected male individuals. So only the females can pass the disease on to affected males. Affected males can only pass the condition on to female members. Recessive inheritance is generally due to 2 faulty genes, and the combined severity of these 2 mutations will determine the severity of the disease. So if you have 2 severe mutations, you get a severe disease. 2 mild mutations, you get mild or even no disease manifesting. You have 1 severe, 1 mild, you get moderate disease. So the condition can manifest in many different ways based on the severity of the disease. But the inheritance is pretty straightforward. So here you have both parents who are carriers who are not affected by the condition, but their children has a 1 in 4 chance of inheriting both faulty copies. The most complex of all is actually the dominant pedigree. So these mutations can lead to loss of function, gain of function, or dominant negative effects. So when there's a loss of function, it means that 50% of what's produced is not enough in the eye. Often we find these mutations in proteins, in genes that code for proteins that can be used all throughout the body, but yet the eye is only the only organ that's affected. So for most parts of the body, 50% is enough, but for the eye, it's not enough. There are also mutations that cause increased function. For example, if there's duplication of the gene or if the enzyme activity is increased, it can be toxic. PRPS1 is one example of that. And then a mutation that are causing inhibitory effects, for example, PRPH2, when the mutation can cause one copy to make a protein, which then binds the healthy copy's protein, which is normal, then that has an inhibitory effect on the healthy copy's product protein. So here where it gets very complicated. So a simplex case where there's only one person affected in the family can be due to a recessive gene, can be due to X-linked gene, can be a dominant gene. So we can't assume when there's only one person in the family that this is a recessive condition, which is what people tend to assume it is. Dominant gene occurs in this situation when the patient's adopted, so we have no access to parental information, when there's a de novo mutation, or when the parents are non-penetrant. Then there are also situations where the dominant disease can present with a recessive pattern, and where a recessive disease can present as a pseudodominant pattern. I will explain a little bit more. So pseudo-recessive, you can see this pedigree where neither parents are affected, but there's 2 children affected here, so you assume this is recessive. But without further information, you don't really know that, because here the father is actually affected. But we may not know that, because if the father died young, he may have never manifested the disease. So you don't really know prior information. And if we do genetic testing, we might find that this patient is a non-penetrant carrier. And in fact, there's 2 other non-penetrant carriers. So you can have a dominant disease, which looks like a recessive pedigree. Here's a pseudo-dominant pedigree. You have the mother affected and 2 children affected. You think this is dominant, pretty straightforward. But then when you do the genetic testing, you realize actually the father is a carrier of a mutation. And this is often comes up when you have the siblings with very different manifestation, because one of the copy of the mutation is from the father, one's from the mother, but the mother has 2 copies, and they could be mild and severe. So you get different manifestation of this. So a dominant pedigree doesn't always means it's a dominant disease. So in our Retina Australia grant, we found 57 families with what we presume was dominant inheritance based on history. So this is the key point. Just because the history tells us it's dominant doesn't mean this is a dominant gene. The probands are current age of 50. It ranged from 11 to 89. Symptom onset from about 25 years of age, but that's a huge range. We have 41 pedigrees with preliminary results. 10 are still pending analysis at Molecular Vision Laboratory in the US, and 6 patients have not provided DNA. And so there's always a proportion of patients who are not willing or not wanting to know their genetic diagnosis, and we leave them as they are. And at some stage, they may change their mind, or that their children wants counseling regarding their risk of passing on the condition, then they might come back to ask for genetic analysis. There were 3 other families who presented with simplex or pseudo-recessive inheritance who were found to have dominant mutation. So here are the combined, or here the results from the dominant families. When we get the report back, you send a DNA off, you get a report that says it's positive, likely positive, or inconclusive. 63% of the report says have a result that's likely positive or positive, but quite a number of them have inconclusive results. If you just send the DNA as a swab to these companies, you're stuck there with inconclusive result in 37%. In fact, even the 63% is not that conclusive, because you really need to test the variant in other members of the family in the affected and the non-affected family to be absolutely sure. So further analysis needs to be performed on not just the 37%, but also those 63%. With further analysis, we were able to solve 7 additional families in this inconclusive basket. 2 are still undergoing further testing of suspected causative gene, which was listed as inconclusive. And we're left with 6 families, which we have no idea still what the genetic cause is. And they're clearly dominant inheritance with the RP. So here we have 63% who have positive results based on just the report alone, but that requires confirmation on segregation analysis. So that's 2 extra samples of DNA to be sent off for analysis. And then with the Australian IRD registry team's help, expertise in this, and further testing for large deletions, which wasn't covered in the previous panels, we managed to solve another 22%. So we're now with 85% of the families solved. Here are the breakdown of the various families. So by far the most common is RP11, and there are 10 families in WA, which affect 52 individuals, and I've examined 30 of them. It's actually very difficult to examine all of the affected family members, and the reasons are either they've passed away, obviously we can't examine them, or their history of we get told about these individuals, but there's just no access to these people, or they don't want to come in for examination. Rhodopsin is second most common. It causes RP4. We have 30 affected individuals in these pedigrees. And then there's RP1, 4 families, 16 affected individuals. HK1, which is hexakinase 1, 3 families, separate families, all with the same mutation, but they're not related. And PRPH2, 2 families. And then you can see a scatter of other genes that only occurs in 1 family, RP9, RP65. You know, RP65 is a recessive disease, but it can also cause dominant disease. SAG typically is a recessive disease as well, but it can also cause dominant RP. And similarly, some of these other ones can be recessive or dominant, but this is all dominant RP. So with focusing down on PRPF31, we have 10 of these, 7 presented with a dominant pedigree, so it was straightforward to know this was dominant disease, but 3 had simplex or pseudo-recessive pedigree. And the reason was that either they were adopted or there was non-penetrance in the family. So one of the parents had it, but wasn't manifesting the disease. 7 of them were found on the panel as positive or likely positive, but 3 were inconclusive, and we had to search for the mutation or deletion by doing further testing. And 3 of the 10 families had members in the family who were carriers of the mutation, but not manifesting RP. And it's been reported that less than 5% of these mutation carriers don't have the disease. You can look at it in the eye very carefully, do electrophysiology, and they're normal. They don't have the disease, and yet they have the mutation in the eye cells. And the way it happens is because the healthy copy of the PRPF31 can compensate the reduction in the faulty copy function by overproducing PRPF31 protein. And because of this understanding and also our understanding of how this gene is regulated, we were able to develop a treatment for PRPF31. We targeted C03, which is another gene that suppresses PRPF31. So if we can suppress the negative regulator, that will allow more of the PRPF31 on the healthy copy to be produced, and this will then overcome the deficiency. So 2 years ago, I presented this timeline. We had the ward study started in 2015. There was a collaboration between Lehigh Murdoch University, between myself and Sue Fletcher. We had PhD students working on this, which led to the discovery of this invention. Then the collaboration with PYC to further progress in preclinical studies. And that today indicates the talk 2 years ago. We had a plan to get this drug through the pipeline to get FDA approval, looking at rabbit, monkey studies, large animal studies, and a plan for clinical trial this year. So this is the timeline now in 2022. So we're almost there at the completion of the preclinical testing. The clinical trials planned for next year. Once we submit the FDA for approval, it can then be used in human. What we'll do prior to that also is a natural history study. So there are several sites in the US and hopefully in Australia as well, looking at PRPF31 natural history. So that's the update on the most common dominant RP gene in Australia. I presume similar situation is found in other states. And I need to acknowledge Lions Institute, UWA, Murdoch University, and specifically Retina Australia for funding this project to allow us to identify these patients. PYC Therapeutics for their help with the preclinical studies, and all the various funding bodies and donations. Our team of scientists and fellows in working on these cells and recruiting patients to the studies, and Jade for organizing all the meetings between myself and my team. John DeRoche, team IRD registry is critical and indispensable in our work, because without them, a lot of these families will still not be solved, and we'll still be able to diagnose many of the families who had inconclusive results from the initial panel. And clinic staff in looking at these patients, and the referring clinicians, optometrists. Thank you.
Thank you so much, Fred. That was great, and so exciting to hear about these new developments and the upcoming clinical trial. So we've got an opportunity to have some more questions from people. I think Alex is still around. So if you've got any questions for either Alex or Fred, just raise your hand. You can also use Alt-Y on the keyboard if that's more convenient for you. And we'll just wait to hear from people. Has anyone got a question? I was going to ask a question actually, Fred. I just wanted to get an idea. You talked about a panel of genes that you're testing for. How many different genes are in the panel? Because obviously you've shown us some there, but were there a whole lot more that didn't appear in that?
Yeah, so it varies depending on when we test the gene. So in the very early phases, we're talking about like only 50 genes, but now the Oregon gene panel has 988 genes. They also look at copy number variations as well in addition to that. And even with 988 genes being looked for, I don't think the rate of finding positive results is going to increase, because the majority of the new genes that's added to the panel is not actually related to RP genes.
Amazing. What sort of timeline is there on the clinical trial that's starting? How long do you think that process will take?
In general, these trials last several years. There are also phase 1, 2, and 3. So the different trials for different purposes. Phase 1 trial tend to last 1 to 2 years, looking for toxicity and tolerability. And many drugs are knocked out at that stage. So if they cause any side effect, that's not anticipated because they didn't see in monkeys, then it goes no further. Those that pass first phase 1 will go into phase 2. Sometimes they combine phase 2, 3, which can last another couple of years for each patient. For rare diseases, because it takes longer to recruit, you can imagine you're trying to find these patients. It may take 2 years to find all the patients, and another 2 years for the last patient to finish. So you're talking about 4 years from starting, another year for results. So that's 5 years just for phase 2. And then after phase 2, you got phase 3. And phase 3, depending on the design, for drug therapy, usually again 2 years, like intravitreal injections, or usually 2 years. For gene therapy, it's usually 5 years, plus a running of 1 year. So they'll get treated in one eye, another year later, get treated in the other eye, and then follow for 5 years. So that's 6 years for each patient. And if you're looking for rare diseases, it depends on how quickly they can recruit. For rare patients, it may take several years to recruit that.
And how many patients do you need to make a reasonable number?
It depends on the purpose. So again, for early phase, for toxicity, maybe a handful low dose, handful high dose to see whether there's any side effects. So you could be looking at 10 to 15 or 20. Whereas for the larger trials, for gene therapy trials, we're talking usually about 50 patients. It would be worldwide, of course, because it's very hard to find 50 rare disease patients. At different, they'll be randomized in one eye, usually not treated in both eyes. The delay in giving the treatment in those who are randomized to sham will give them some idea whether that treatment is better than placebo.
Sally, there's a question in the chat. If we have family members that haven't sent in DNA, is it too late for them to do so?
Never too late. Never too late. The DNA lasts basically forever. So we generally don't need to collect DNA unless we had to test it many, many times and we used up the material. But those who haven't sent DNA, yeah, they're very welcome to send in.
Yeah.
There's another question here from Kelly. Is there any research currently being undertaken for punctate inner choroidopathy?
That's an inflammatory disease. There may be genetic association, but we're not working on any genetic basis of inflammatory disease at this stage. But certainly, that's an interesting topic. There are researchers who are looking at genetic predispositions to pick disease response to treatments or non-responders or different manifestations of that.
Any further questions? Does anyone have? If there aren't, I might say thank you very much to our speakers and hand over to Leighton Boyd, the chairman of Retina Australia, just to say thank you.
Well, on behalf of Retina Australia, we're very lucky here in Australia. Us as patients, people with inherited eye diseases, we can be unlucky in one way that we have our various eye conditions, but we're in a very fortunate situation that we have lots of teams, and certainly Alex and Fred are two people that have had a long association with Retina Australia. They've presented numerous times themselves and various members of their team. Us in Retina Australia, we welcome all the donations from all of our donors, obviously, and our members, but this has enabled Alex and Fred, in this case, to put together and do some special work on behalf of us as patients. The collaboration across this country is amazing, and as an organisation, our aim is to bring it all together as often as we can and to help us to find a treatment and cure eventually for us all. So thanks to both of you today. It always amazes me when I hear these presentations how such a very, very difficult topic of trying to explain so people like us as lay people can understand it. The analogies that were given, and fortunately Rosemary's here and could explain to me all these graphics and the cakes and all the rockets and all the other things, and it's just makes things easier for us to understand. We are grateful for your time. We're grateful for your work and your teams. And I thank you all on behalf of all of us here. We can do a virtual round of applause, and thank you for your time.
Thanks very much, Leighton.
Just on that, can I just do a sales pitch? People like Alex and Fred can only do their work, and we help contribute a little bit, and they do get bigger grants from NH and MRC and all other sources, and and we as an organisation try to do what we can to help them out. The money that we give is seed funding as an organisation, and we've given over $6 million since Retina Australia started giving out grants, which is, I think, astounding and amazing that all these people online today and people that aren't online we're so grateful about. And on June 21 this year, we will be running our Giving Day. Last year on the same day, that coincides with the winter solstice, we're having our Giving Day. Now, we'll launch that on June 10 this year, and all donations given on that day and after June 10 will be matched by our match donors. Last year, we raised around 78,000 in 24 hours, which is an amazing effort. And I just would like everybody online here today and all your friends and email lists and your contact lists, let people know that this day is on June 21 again this year. And we encourage you all to give, and that will enable us as a board to make decisions and to fund more applicants, more researchers in the next years ahead. So thanks again to everyone, and I'll hand back to you, Sally.
Thanks so much, Leighton. So that's the end of the webinar. Thank you so much to both of our presenters for your time and the wonderful presentations. And thank you all for coming along this afternoon, and enjoy the rest of the day. Thanks so much.
Bye.